Streptococci of particular medical interest. Streptococci of particular medical interest include Streptococcus pyogenes group A, Streptococcus agalacti group B, Enterococci, Verdinous Streptococci, Streptococcus pneumoniae, Streptococcus, and related genera. Streptococcus pyogenes habitat and transmitted or transmission. Normal habitat is the human upper respiratory tract and skin. May survive in dust for some time, spread by airborne droplets and by contact. Characteristics: a common sound and nasopharynx of minority of healthy adults, but more commonly about 10% in children. Grows well on blood agar with a characteristics halo of beta hemolysis. Some strains produce mucoid colonies as a result of having a hyaluronic acid capsule. This may contribute to virulence by offering resistance to phagocytosis. Antigenic structure include, number one, include group specific wall antigen. This carbohydrate is contained in the cell wall of many streptococci and forms the basis of serologic grouping Landsfeld group C substances from A to H, K to U. 2. M protein. This substance is major virulence factor of group A Streptococcus pyogenes. M protein is filamentous structure anchored to the cell membrane that penetrates and projects from the streptococcal cell wall. When M protein is present, the streptococci are virulent, and the absence of M specific type antibodies, they are able to resist phagocytosis by polymorphic nuclear leukocytes. Group A streptococci that lack M protein are not virulent. Because there are more than 150 types of M protein, a person can have repeated infections with the Streptococcus pyogenes of different M types. M protein is thought to serve as virulence factor in two ways: by enabling Streptococcus pyogenes cells to adhere to surface. 2. Functioning as antiphagocytic. 3. T substances. T substance, this antigen has no relationship to virulent streptococci. Unlike M protein, T substance is acid label and heat label. T substance permit differentiation of certain types of streptococci by agglutination with the specific antisera, while other types share the same T substance. Yet another surface antigen has been called R protein. 4. Protein F, fibronectin binding protein. Protein F mediates attached to fibronectin in the pharyngeal epithelium. M protein and lipoic acid also bind to fibronectin. Toxin and enzymes. Streptokinase. Streptokinase is produced by many strains of group A beta hemolytic streptococci. It transforms the plasmogene of human plasma into plasmine, an active proteolytic enzyme that digests fibrin and other protein. This process of digestion may be interfered with by non-specific serum antibodies, inhibitors, and by specific antibody, anti-streptolysin or anti-streptokinase. Streptokinase has been given intravenously for treatment of pulmonary embolism and by coronary artery and venous thrombosis. Type 2 streptodenorase. Streptodenorase depolymerized DNA. The enzymatic activity can measure by the decrease in viscosity of known DNA solution. Mixture of streptodenorase and streptokinase are used in enzymatic debridement. They help to liquefy exudates and facilitates removal of pus and necrotic tissue. Antimicrobial drugs, those gain better access and infected surface recover more quickly. Hyaluronidase cleaves hyaluronic acid, important components of growing substance of connective tissue, into numerous small fragments, thus converting the gel state of extracellular matrix to solid state. The consequence of this reaction is to permit the rapid spread of the infecting microorganism through the connective tissue space, so-called spreading factor. Biogenic exotoxin. Biogenic exotoxin elaborated by group A streptococci. The streptococcal biogenic exotoxin have been associated with the streptococcal toxic shock syndrome and this scarlet fever. Dephosphoridine nucleotides. This enzyme elaborated into the environment some streptococci. This substance may related to the organism ability to kill leukocytes. Proteinase, amylase are produced by some strain. Not all these products are produced by every strain. The combined action of enzymes and toxin contribute to the pathogenicity. Hemolysins. Hemolysine is divided into two. Streptolysine or streptolysine is a protein that hemolytically active in the reduced state, but rapidly inactive in the presence of oxygen. Streptolysine O is responsible for some of the hemolysis seen when grow in acute deeps into the medium in blood agar plates. Streptolysine O combines quantitatively with the antistreptolysine O, ASO, an antibody that appears in human fellow infection with the streptococci that produce streptolysine O. This antibody blocks hemolysis by streptolysine O. This phenomenon forms the basis of a quantitative test for the antibody. An anti-streptolysine O ASO serum titer in excess of 116 to 200 units is considered abnormally high and suggests either recent infection with the streptococci or persistently high antibody levels due to exaggregated immune response to earlier exposure in hypersensitivity person. Streptolysine S. Streptolysine S is the agent responsible for hemolytic zones around streptococcal colonized growing on the surface of blood agar plates. It is elaborated in the presence of serum, hence the name streptolysine S. Culture and identification. Culture on blood agar elite characteristics beta hemolytic colonies. Lysis of blood due streptolysine O and S. A gram stained smear show gram-positive cocci in chains, which are well developed in liquid rather than solid media. This isolate can be presumptively identified as Streptococcus biogenes if it is sensitive to bastracine. If rheumatic fever is suspected, then testing the patient anti-streptolysine O ASO antibody titer will demonstrate previous exposure to Streptococcus biogenes. Streptococcus biogenes cause a number of infections. The most notable are tonsillitis and pharyngitis, peritonsillar abscess, scarlet fever, mastitis and sinusitis, wound infection leading to cellulitis and lymphangitis, impetigo, invasive group A streptococcal disease, streptococcal toxic shock syndrome. We can Note that streptococcus pyogenes cause number of infection like tonsillitis or pharyngitis, like what you see now. If the disease combined with the lesions or yellow lesions is considered bacterial infection, while if lesions combined with the red lesions consider viral infection. Complication after episode of infection, some patients develop complications such as rheumatic fever, glomerulonephritis, and erythema, which may have long-lasting effects. Note, in cellulitis, hyaluronidase mediates the substance spread of infection. Erythrogenic toxin cause rash of scarlet fever, post-streptococcal infection manifesting as a rheumatic fever. This automatic disease occurs 2 to 3 weeks after the initiation of pharyngitis. It is caused by cross-reaction between antigens of heart, joint, tissue, and nervous system, and the streptococcal antigen, especially M protein epitopes, due to immune response against streptococcus antigens similar to heart and joint tissues antigens. It is characterized by fever, rash, carditis, and arthritis. Rheumatic fever, autoimmune disease, damage to tissue after streptococcal infection. Each year, 470 cases per year associated with the streptococcal pharyngitis, etc. B. Acute glomerulonephritis is caused by immune complex bound to glomeruli, disposition of antigen-antibody complex, activation of complement. Treatment and prevention. Streptococcus pyogenes has not developed resistance to penicillin. So penicillin remains the drug of choice to treat streptothroat and most other streptococcus pyogenes infection. Some strains have developed resistance to erythromycin and many strains are resistant to tetracycline, no vaccine available. In penicillin allergic patients, macrolides such as clarithromycin or azithromycin is the preferred drug. Penicillin G plus clindamycin used to treatment necrotizing fasciitis and streptococcal toxic shock syndrome. Clindamycin is added to penicillin to inhibit protein, that is toxin synthesis, so that a huge amount of toxin is not released abruptly from rapid dying bacteria. Streptococcus agalacti group B, habitat and transmission, the human vagina and sometimes anorectal carriage. Babies acquire infection from colonized mother during nursing, common in cattle and cause bovine mastitis. Characteristics: gram-positive cocci in chain, catalase negative organism. Gram smear and culture, which yield B hemolysis colonies on a blood agar. Colonies in a blood agar are generally larger colonies and less hemolysis than group A streptococcus pyogenes. Landsfeld group determined by antisera against cell wall polysaccharide. Virulence factor. Encapsulated strain of GBS are resistant to phagocytosis by white blood cells. Other potential virulence factor include hemolysins, CAMP factor, peptidase, and hyaluronidase. Pathogenicity. cause neonatal meningitis and septicemia, also associated with the septic abortion and gynecological sepsis. Treatment and prevention. Penicillin and ampicillin are the antibiotic of choice. Erythromycin in patients hypersensitive to penicillin. In life-threatening infection, aminoglycoside can be added to regime. Note, pregnant carriers should be treated with the ampicillin during labor if risk factors such as premature rupture of membrane or prolonged labor are present. Interpartum prophylaxis of group B streptococcal carries and administration of antibiotics to their newborns reduce neonatal group A, group B streptococcal sepsis by as much as 90%. Enterococci. Enterococci, this belong to Landsfeld group D and include Enterococcus faecalis and other less important species. Habitat and transmission, normal intestinal inhabitants. Culture and identification. Most strain non-hemolytic grow rapidly on bile-containing media, example McConkey medium. Their ability to grow in 6.5% sodium chloride, which is 7 times the concentration found in normal tissue fluids. Pathogenicity. Most commonly cause acute infection of the urinary tract infection and sometimes wound infection, especially after intestinal operation. Treatment, antimicrobial susceptibility test is very important for enterococci. Verdun streptococci. This is mixed group of streptococci with the variable characteristics. Hence, the nomenclature of this group is in constant state of flux. Typically, they are alpha hemolytic, but they may be non-hemolytic. These streptococci principally live in the oropharynx. Oral streptococci can be divided into 4 main species group as follows: Streptococcus mutans group, Streptococcus salivarius group, Streptococcus milleri group, Streptococcus oralis group. Habitat and transmission. The emergence group of streptococci are common inhabitants of the oral cavity and comprise highly 1/4 of the total cultivable flora from supravaginal and supragingival and gingival plaque and 1/2 of the isolation from tonic and saliva. They are vertically transmitted from mother to child. Infection, endocarditis caused by the virulence group is generally a result of their entry into the bloodstream during intraoral surgical procedure, example tooth extraction, and sometimes even during tooth brushing. Culture and identification. Gram-positive cocci in chains. Alpha hemolytic, catalase negative, growth not inhibited by bile or optochain. In contrast to pneumococci, commercially available kits are highly useful in the laboratory identification of this organism, example EPI kit system. Pathogenicity. The major agents of dental caries in Streptococcus mutans. They have a characteristic ability to produce voluminous amounts of sticky extracellular polysaccharide in the presence of dietary carbohydrates. These help tenacious binding of the organism to animal and to each other. Streptococcus mutans may act as opportunistic pathogen under a number of circumstances. A breakdown of the dental enamel from the acidic fermentation products in the development of the caries lesion result in invasion of dentine by the microorganism and eventually in bulb infection. Streptococcus mutans has been isolated from the root canals of such teeth but at low incidence. It has been demonstrated experimentally that Streptococcus mutans can destroy cervical bone when it is inoculated into the dental bulb, and that same organism is isolated from blood as late as 21 days after the inoculation. They are also important agent of effective endocarditis, usually bacteria released during dental procedures, still on damaged heart valves, causing inactive endocarditis. And not in Streptococcus mutans. Streptococcus mutans gained notoriety in the 1960s when it was demonstrated that caries could be experimentally introduced and transmitted in animals which were orally inoculated with the organism. The name mutans result from it is a frequent transition from coccal phase to coccobacillary phase. They are currently 7 distinct species of human and animal mutant streptococci: Streptococcus circus, Streptococcus downi, Streptococcus feras, Streptococcus macaci, Streptococcus mutans, Streptococcus ratas, and Streptococcus sabrinus. The term Streptococcus mutans is limited to human isolates belonging to 3 streptococci C, E, and F. Treatment: prophylactic therapy against endocarditis resulting from Streptococcus mutans probably is most effective with the use of ampicillin combined with the gentamicin. The organism is also sensitive to tetracycline, vancomycin, erythromycin, chloramfenicol, but it is resistant to cephalosporin and streptomycin. Streptococcus pneumoniae. The pneumococci are gram-positive diplococci, often lancet-shaped or arranged in chains, possesses a capsule of polysaccharide that permits typing with the specific antisera. Pneumococci are differentiated from many species of oral streptococci by being sensitive to optochain and syllable in bile. Pneumococci are normal inhabitants of upper respiratory tract of 5 to 40% humans. Streptococcus pneumoniae is the most common cause of community-acquired pneumonia and adult bacteria meningitis, and is important cause of otitis media, sinusitis, and mastocytitis. The risk of disease is highest among young children, older adults, smokers, and persons with certain chronic illnesses. Like figure. Streptococcus pneumoniae culture. Pneumoniae form a small round colony at central plateau with the elevated rim. Pneumococci are alpha hemolytic on blood agar, growth enhanced by 5 to 10% CO2. Quinine reaction is used to determine Streptococcus pneumoniae. Pathogenesis. A. Production of disease. Pneumococci produce disease through their ability to multiply in the tissue. They produce no toxin of significance. The virulence of the organism is function of its capsule, which prevents or delays ingestion or phagocytosis. A serum that contains antibody against the type-specific polysaccharide protects against infection. B. Loss of natural resistance. Since 40 to 70% of humans are at sometimes carries of virulent pneumococci, the normal respiratory mucosa must possess great natural resistance to the pneumococcus. Among the factors that properly lower this resistance are those predisposed to pneumococcal infection are the following. Number 1, abnormalities of the respiratory tract. Viral and under infection, damaged surface cells, abnormal accumulations of mucus, example allergy, which protects pneumococci from phagocytosis. 2, alcohol and drug intoxication, which depression phagocytic activity, depresses the cough reflexes and facilitates aspiration of foreign material. 3, abnormal circulatory dynamics, pulmonary congestion, heart failure. Other mechanism, malnutrition, general debility, skull cell anemia, hypocyplanism, nephrosis, or complement deficiency. Treatments: Streptococcus pneumoniae, isolate were high sensitive to penicillin G, the initial agents of choice, until the last 1918s. Since then, the incidence of penicillin resistance has been increased worldwide. Most strains remain sensitive to third generation cephalosporins such as cefotaxime, ceftriaxone, and all are still sensitive to vancomycin. These antibiotics are therefore the agents of choice for invasive infections by penicillin-resistant strain of Streptococcus pneumoniae. Streptococcus and related genera. Streptococcus and related genera, these streptococci grow only under anaerobic or microaerophilic conditions and variably produce hemolysins. They are part of the normal microbiota of the mouth, upper respiratory tract, bowel, and female genital tract. They often participate with the many other bacterial species in mixed anaerobic infections. Such infections may occur in wounds in the breast, in postpartum endometritis, after rupture of abdominal viscus, in the brain, or in the chronic suppuration of the lung. The pus usually has a foul odor. Thank you for listening.